Hello and welcome to my first video on my channel, and today we're going to be talking about cells. It is important to stress, however, that every cell is unique, it's different, and therefore that we're just going to be focusing on stereotypical animal, plant and bacterial cells. So without further ado, let's begin. So here, for those of you who don't know, I've drawn up a stereotypical animal cell and labelled some important organelles within that cell. So the process of splitting up a cell, such as the animal cell, into different organelles is known as compartmentalization. Now each of these organelles plays a particular role, which we'll come on to just in a little bit. Now we also have the plant cell. So you can see there's some instant differences between the plant cell and the animal cell. So one of the main differences is the presence of this cellulose cell wall. Now this cellulose cell wall is present within a plant cell, but it's not present within an animal cell. Now we can focus on the structure and the function of these organelles that we've just had a look at here within the animal and the plant cell. So the structure and the function of specific organelles. So here we have the nucleus. So what the nucleus does is it's possibly the most important organelle within a cell. So this controls the activities of the cells via the production of mRNA, which is just an abbreviation for messenger RNA, something which we'll come on to in a later video, but just a heads up, it is involved with protein synthesis. The nucleus is also involved as it contains the genetic material in the form of DNA coiled around histone proteins to form linear chromosomes. Now, the nucleolus is what's known as this little black dot here I've drawn within the outer nucleus. So the nucleolus is used to manufacture and assemble rRNA, and this assembles ribosomes. So just a quick note as well that the nucleus has an outer membrane, it has a double membrane structure. We refer to this as a nuclear envelope. Now it's also important to note that there are little tiny holes within this nuclear membrane, and this is again involved in the process of protein synthesis, which we'll study in a later video. The mitochondria. So, the mitochondria are small rod-shaped organelles which are typically between 1 and 10 micrometers in length. So, these too have a double membrane, and this is known as the mitochondrial envelope. The inner membrane folds to form a cristae, which provides a large surface area for enzymes to attach to in the process of aerobic respiration to produce ATP or energy for the cell. So the outer membrane is what controls the movement of substances into and out of the mitochondria. Ribosomes. Ribosomes are a site of protein synthesis, and in larger cells such as eukaryotes, which are just cells which have the DNA located within a nucleus, these ribosomes are usually large. The Golgi apparatus is an organelle which modifies and packages proteins into vesicles. Once proteins are assembled within protein synthesis, they are packaged and sent off to the Golgi apparatus for modification. So this modification could be the addition of addition of iron or non-protein portions, which are known as prosthetic groups. So we then move on to the rough endoplasmic reticulum. The rough endoplasmic reticulum is continuous with the nuclear membrane, and the process of this is to provide a large surface area for protein and glycoprotein synthesis, and it also provides a pathway for transporting proteins and other materials through the cell. Now, if the rough endoplasmic reticulum is involved with proteins and glycoproteins, the smooth endoplasmic reticulum is involved with lipids and carbohydrates. So the smooth endoplasmic reticulum is used to synthesize, store and transport both lipids and carbohydrates throughout the cell. So we move on to a few other organelles. So a vesicle, the main process of a vesicle is involved with transport around the cell. So the vesicle are just a small fluid containing membrane-bound organelle, and another role of the vesicle is involved in phagocytosis. The vacuole. Now the vacuole is present in a majority of plant cells, but it can also be present in animal cells. The vacuole is used to keep the cell turgid. So what that means is just provide structural support to the outside of the cell. It's important to note that the membrane of the vacuole is known as the tonoplast. Now the tonoplast is important because it separates the contents of the vacuole from the cytoplasm and all other organelles on the outside. Now the cell wall, like I've mentioned before, is only present in plant cells. So this cell wall is made of cellulose and provides structural support for the cell, prevents the cell swelling when it is placed in a high water potential. Cell membrane is present in both animal and plant cells, and the role of the cell membrane is to control the movement of substances into and out of the cell. Lysosomes are spherical membrane-bound sacs that are produced by the Golgi apparatus. They are typically 1 micrometer in diameter and have an approximate pH of around 5. The function of lysosomes is to digest unwanted materials in the cytoplasm, and therefore they have an important role in the process of phagocytosis. Finally, chloroplast. So chloroplast is a complex organelle that's within the cell, and it has 3 layers. So the outer layer is known as the outer chloroplast membrane, and this is an outer layer that allows small molecules and ions to easily diffuse into the chloroplast, but it cannot allow large protein molecules. The inner chloroplast membrane is continuous with a fluid-filled centre, which is known as the stroma. This controls the passage of materials into and out of the stroma. It is also a site of fatty acids and lipid synthesis. The inner membrane is then continuous with the thylakoid system. Finally, the thylakoid system is the particular location of photosynthetic pigments, which are found embedded in thylakoid membranes. So, with it short, the chloroplast is involved in photosynthesis because it contains photosynthetic pigments, which are known as chlorophyll molecules. So we've focused both on eukaryotic cells, which are as I said previously, cells which contain their DNA surrounded by a nucleus. However, they're not the only type of cells which exist. We have prokaryotic cells, such as this bacteria cell, which does not have its DNA located within a nucleus. This has its DNA in the form of circular DNA, which lies free in the cytoplasm. In other words, it contains non-membrane-bound organelles. And as you can see, there's some few differences between the bacteria cell and the plant and the animal cell that we have just focused on. Now, we will finish off with the structure and the function of the bacterial cell's organelles. So the bacterium cell, you may have noticed, has a flagellum. And the function of this flagellum is to provide movement. It has a small motor at the base of the flagellum, and it is used to provide movement to the cell. Circular DNA is the main store of DNA main store of genetic information within the bacteria cell. So the bacteria cell, like I've just said, is a prokaryotic cell and does not contain its DNA or genetic information within a nucleus, and therefore it lies free in the cytoplasm. The cell wall is made of different substances to that of the plant cell wall, and however, it does conduct the same function to provide structural support to the cell. The pili are protein hair-like structures which extend from the cell membrane and the cell wall, and they have a role in cell adhesion and cohesion. The cohesion to other bacterial cells and adhesion to other substances. The plasmid or the plasmids in a bacterial cell contain extra DNA or extra genetic information which helps the bacterial cell to survive. So the mesosome are what's known as invaginations in the cell wall. Now, many scientists have debated what the function of this organelle actually is, and many believe it is just some scientists have predicted that it is involved in aerobic respiration, but the general consensus is that it is merely just an invagination in the cell wall. Now, finally, we have ribosomes. Ribosomes, just like in eukaryotic cells that we've just focused on, are for a process of protein synthesis. However, bacterial cells are prokaryotic, and therefore their ribosomes are often a lot smaller, but they do conduct the same role for protein synthesis. Now, thank you for watching this video. I hope you've got a lot out of it. We've learned all about animal, plant and bacterial cells, and hopefully you've learned something about the structure and the function of the organelles within these cells. Thank you very much for watching, and it would be very helpful if you could like, subscribe and leave a comment down below. Thank you, and I'll see you again soon. Goodbye.